Hello, and welcome back to The Sound of Science. Today, we're going to be talking about mitosis and the cell cycle. I know that says meiosis up there, and we will talk a little bit about meiosis, but mainly just to give you a definition. So, don't get too hung up on that yet. We're going to cover more of that when we start doing genetics in the next unit. The first question is, why do cells divide at all? If cells get too big, then their surface area to volume ratio gets off. And they're going to have issues such as traffic and information, which sounds a little bizarre, but think of it this way. Remember in the last video, we talked about the surface area to volume ratio, and I showed you guys those gelatinous cubes. And inside that gelatinous cube, there was an area that couldn't be reached by things from the outside of the cell. And I told you that the larger the cell gets, the more of that area can't be reached. Well, that's what we mean by traffic or information. It's the signals, the materials that need to get to the inside of the cell can't get there if the cell is too large. So once the cell reaches a certain point in size, it's actually going to divide. It is actually a trigger that will cause it to divide so that isn't a problem. That doesn't happen. Cells may also need to reproduce asexually. Cells may need to create those specialized reproductive cells. This is the process of meiosis. I told you you need to know the definition of it, but not really the steps. So meiosis is just the creation of specialized reproductive cells. Cells may need to be replaced to heal damage, and cells may need to be increased so that an organism can grow. As we go through the steps of the cell cycle, one of the things that you need to be aware of is that there are regulatory proteins that are controlling this whole process from both inside the cell and outside the cell. Basically, there's checkpoints. The cell cycle can't proceed unless certain conditions are met. And this is actually incredibly important. At the end of this video, we're going to talk a little bit about where cancer cells come from and its problems with these checkpoints and these regulatory proteins that lead to cancer. So why do we have so many steps of the cell cycle, which I know I haven't shown you yet, but you're about to see? And why does cell division have to happen in such a regimented way, such an organized way? And really, that's because we're trying to keep chromosomes organized. Remember, chromosomes are incredibly important. The cell can't do its job without the DNA. Any issues with the chromosome can lead to a major genetic issue or even lead to cell death. So you also have to remember, too, that we have different types of cells, prokaryotes and eukaryotes. So prokaryotes typically are just going to have a single circle of DNA known as a plasmid. And as a result, their cell cycle is a little bit simpler, a little bit easier. Eukaryotes have lots of chromosomes. Those chromosomes are basically DNA that's been wound up really tightly. It's controlled with these histones that bind it. And basically, that's how the DNA turns into a chromosome. Typically, DNA is going to stay loose. We call it chromatin in that stage until it's time to divide, and then it forms into chromosomes. The cell cycle in prokaryotes is ridiculously simple. Basically, they're going to make a copy of their DNA and they divide. And this can happen as often as every 20 minutes. Now, remember, prokaryotes are all bacteria. Bacteria are prokaryotes. That's all the prokaryotes that exist are bacteria. So bacteria can actually reproduce every 20 minutes. This is their type of reproduction. This is asexual reproduction for them, and it's known as binary fission. So just remember this. Keep this in mind when you decide that, something falls on the floor and you're like, oh, 10-second rule. It's fine. There is no 10-second. Okay. Let's talk eukaryotes. Obviously, this is a lot more complicated and a lot more complex than what we typically will see with a prokaryote, right? There is so much more going on here. Eukaryotes, when they go into their cell cycle or as part of their cell cycle, they're actually going to have a G1, an S, a G2, and an M phase followed by cytokinesis. All of this is part of what cells do all the time. So we're going to go through each of these. I'm going to talk about each of these, and hopefully that will help you sort of understand what your cells are doing. Okay, so now that I've cleaned this up a little bit, let's take a look. G1, or gap phase, basically what the cell is going to do is it's going to grow, it's going to make new proteins, and it's going to make its organelles. S phase is our synthesis phase. This is where DNA replication is taking place. DNA is going to be copied. The cell at this point is going to contain twice as much DNA as it needs. G2, or gap phase 2, makes the organelles that are needed for cell division. And then this is followed by the mitosis phase. Mitosis is actually when the division of the nucleus is going to occur. And then finally, we're going to end with cytokinesis, where the cell is going to split. The G1S and G2 phase, all of this we actually refer to as interphase. Interphase is what the cell does most of its time. If we were talking about a 24-hour period of time, 23 hours would be spent in interphase, and the last hour only would be spent in mitosis. Cells just do interphase most of the time. That's cells just living their best cell life. So keep in mind that mitosis is not something that happens constantly as far as the cell's life goes. It's really going to be a case of interphase most of the time. Okay, so let's talk mitosis a little bit. There are 4 phases to mitosis, and I'm going to go through each of these phases with you. So this is after the cell has gone through interphase, and it's now ready to divide. It's made copies of all of its DNA, it's made copies of the organelles, and it has reached the point where it's created all the spindle fibers, centrioles, all the things that it needs so that it can divide properly. So prophase is the first phase of mitosis. This is when genetic material is going to condense so it becomes visible. We also see chromosomes appearing as sister chromatids. So this is when our DNA goes from looking like a hairball to looking like chromosomes, like we would expect. The nuclear membrane itself is going to start breaking down, and the centrioles are going to produce those spindle fibers or finish producing those spindle fibers so that they can be used to direct the chromosomes to the correct place. This is the first phase of mitosis. Okay, metaphase. Metaphase, I always remember because metaphase means that the chromosomes are lining up in the middle. So remember that prophase is first, metaphase, they're in the middle. The centromeres of the chromosomes line up. And what you see here in the picture is that the spindle fibers are attaching to the chromosomes or to the chromatids. They're being directed by the centrioles. And again, this is showing you in an animal cell. This is an animal cell vision of mitosis. Plant cells are a little bit different. Anaphase, the chromatids are actually going to split apart and they move away. So prophase is the first, metaphase, they're in the middle, anaphase, they move away from each other along the spindle fibers. This is getting close to the end of mitosis. The cell is going to start actually pinching in the next phase, the telophase. Telophase to me looks a little bit like a butt. The chromosomes are going to move all the way apart. Now they're in their individual sections of the cell. The cell starts to pinch in. The nuclear membranes begin to reform. And as soon as they start to reform, the chromosomes actually start to unwind and loosen up, which is important because as soon as the cell becomes 2 cells, it's got cell things to do. So it's going to need to get access to that DNA. Cytokinesis, kind of mitosis, kind of cell cycle. It's at the very end, the cells are completely split into 2 new daughter cells. Why are they called daughter cells and not son cells? I don't know. I think maybe people were like, sun cells? That sounds like fun. I have no idea, guys. But they call them daughter cells. And the really cool thing is they're identical. So when you go through mitosis, the cells, as they are reproducing, they're making exact copies of themselves. They are clones of each other. So these two cells will be identical. Think about what that means for people, right? You actually started out as a single cell, and every single cell in your body is the result of mitosis, which means every single cell in your body has identical DNA. It's absolutely mind boggling. And the reason that your skin cells are different from your eye cells, which are different from your nerve cells, which are different from your muscle cells, is because certain pockets of DNA are being used, or certain aspects of your DNA are being used in some cells and not being used in other. So, just something to kind of keep in mind as we move forward. I absolutely adore this picture. This actually shows you all the phases pretty much in order. So if you take a look, the very first picture at the top, which hopefully you can see my pointer right here, this is what a cell is going to look like in interphase. Interphase. This is the cell just being a cell. And how can we tell? Well, the DNA sort of looks like a hairball in the nucleus. If we move over here, we're actually going to see a cell in prophase. And we can tell it's prophase because our DNA is starting to condense. Our nuclear membrane is starting to fall apart. This is metaphase. And again, how do we know? Because the DNA, the chromosomes are lined up in the middle. This is going to be anaphase. In anaphase, our chromosomes are starting to move away from each other, so it's anaphase. As we move over here, this one and going into this one, these are both going to be examples of telophase, where the cell itself is starting to reform the nuclear membranes around each new set of DNA, and we're starting to pinch in. And then finally, we get to the point where we're achieving cytokinesis, which is sort of hidden under my picture. So we're back to where we started. So this is all of the steps of the cell cycle. So hopefully that sort of helps you understand what's happening there. Okay, special note. Cells actually go through something called apoptosis, and this is planned cell death. So a series of steps will take place, including the chromatin shrinking and basically the cell breaking open. The cell membrane will break open to kill the cell. Neighboring cells then kind of clean up the remains, and that's it. So when your cells get damaged or they get worn out, your cells will literally go through this apoptosis. And this is how we get rid of those cells that really just need to be replaced. As you get older, more and more of your cells are going to go through apoptosis, and less of them are actually going to be replaced. This can also happen with some of the autoimmune diseases you can have, such as AIDS, or autoimmune disorders, or things like Parkinson's. Your cells actually go through apoptosis more than they should. Cancer is what's going to happen when cells don't respond to signals to stop dividing. So, like I said, cells most of the time they're hanging out in interphase. They're just doing what cells do, living their best cell life. But sometimes the cells no longer understand the signals that say stop dividing and go into interphase. And this is when we get tumors, and it's considered malignant when it spreads from the damaged cells or the cancerous cells into healthy cells, and begins to damage tissue. So usually this is going to be caused by a defect in the DNA. It can be completely random, or it can be due to a carcinogen like smoking, UV light, radiation, or even some type of viral infection can lead to cancers. So just kind of something to keep in mind, that cancer is literally when your own cells start acting up. Well, that's all we have time for in this video. And this will conclude our section on cells. So make sure that you go back, you get any of the notes that you might need. Make sure you know all of the steps of the cell cycle. Make sure you know why the cells go through the cell cycle. And make sure you know what each of the phases of mitosis looks like so that you can identify them should you see them again, for instance, on a test. Look forward to seeing you next time.